You went to bed on time, slept 7 hours, woke up, and your brain didn't clean itself. That's not a metaphor. Your brain runs a physical cleaning system every night, and for most adults over 60, it's working at a fraction of what it should. Not because of how long you sleep, because of what type. A 2025 clinical study measured Alzheimer's proteins, beta-amyloid and tau, in participants' blood before and after sleep. The strongest predictor of how much was cleared overnight was not total sleep hours, it was deep NREM sleep duration, the stage adults over 60 lose first and lose fastest. In the next 18 minutes, I'll show you exactly what's happening inside your brain during those lost deep sleep hours and 3 biological changes you can make tonight. Let me describe exactly what happens inside your brain in the first 90 minutes after you fall asleep. Your blood pressure drops, your breathing slows, your muscles relax, and then something begins that took 15 years of neuroscience to properly document, but takes about 30 seconds to understand once someone actually explains it. Your arteries start pulsing differently, not faster, not harder, slower, and in a very specific rhythm. And as those arterial walls expand and contract in that slow pattern, they act like a biological pump. They push cerebrospinal fluid, the clear, colorless liquid that surrounds your brain and spinal cord, through channels that wrap around every blood vessel in your brain. Those channels are called paravascular spaces, and they form the infrastructure of a system that for most of human history nobody knew existed. As that fluid moves through your brain tissue, the way water moves slowly through a very dense sponge, it picks up the metabolic waste that built up during 12 hours of thinking, deciding, remembering, reacting. The proteins, the byproducts, the debris of a brain that was working all day, including 2 proteins you've almost certainly heard of, beta-amyloid and tau. The same proteins found in abnormal concentrations in the brains of people with Alzheimer's disease. Your brain cleans them out every night, or it's supposed to. This system was first described in detail in 2013 by neuroscientist Maiken Nedergaard and her team at the University of Rochester. They named it the glymphatic system, from the glial cells that control its channels and the lymphatic system it functionally resembles. For decades before that, the brain was considered the one organ in the body without a dedicated waste clearance system. Every other tissue had lymphatic vessels doing that job. The brain, scientists assumed, simply didn't need them or had found another way. It turned out the brain had engineered something far more elegant, a system that piggybacked on the very act of deep sleep to run its own maintenance cycle. The moment you stop generating slow waves, the pump slows. The moment your sleep fragments, the cleaning stops. And the discovery changed everything about what scientists think about what sleep is actually for. Not sleep as rest, not sleep as consolidation, sleep as physical maintenance. Sleep as the window during which your brain runs its own sanitation department. Here's the critical part, and this is where most sleep content fails you. This system does not operate the same across all sleep stages. It has a window, a biological window that falls almost entirely within deep NREM sleep, stage N3, also called slow-wave sleep. When that window is shortened, fragmented, or lost altogether, the fluid still moves, but the clearance efficiency drops significantly. My father had his first stroke at 68. In the years before it happened, he slept badly, woke at 2, 3 in the morning, light, restless sleep that left him tired no matter how long he stayed in bed. His doctors adjusted his blood pressure medication, checked his cholesterol, monitored his glucose. Nobody, not a single physician, asked about his sleep architecture. Nobody asked how much of his night was deep sleep, Nobody asked whether the part of sleep that clears the proteins linked to stroke and dementia was actually happening. I've spent years trying to understand what we missed. And what the research now confirms is this: the system that was supposed to protect his brain every night was running at a fraction of its capacity, not because he wasn't sleeping long enough, because he wasn't reaching the depth that clearance requires. Here's the gap that matters. Your doctor can measure your cholesterol at every annual visit, blood pressure, glucose, kidney function.
But not a single routine test in standard medical care tells you how much slow-wave sleep you're generating, or whether your glymphatic system is running efficiently or silently failing. That gap between what the research shows and what actually gets assessed is where the risk lives quietly for years before it becomes a diagnosis. If nobody has ever explained this to you before, you're not alone. Subscribe to this channel. Every video exists to close exactly this kind of gap. Let's talk about what actually happens to slow-wave sleep as you age, because understanding the mechanism of the problem is the only way to understand why the solutions actually work. In young adults, early 20s to mid-30s, stage N3, deep slow-wave sleep accounts for roughly 20 to 25% of total sleep time. In a 7-hour night, that's 90 to 100 minutes of deep, physically restorative sleep, during which the glymphatic system is running at full capacity. By the time you're in your 60s, that number has fallen to 5 to 10%, 35 to 50 minutes on a good night, sometimes considerably less. By 70, some individuals register almost no measurable N3 on a clinical EEG at all. Now, and this is important, the total hours of sleep often don't change dramatically across these decades. Many adults over 60 sleep 7, 8, even 9 hours. The quantity looks adequate on the surface. What changes is the architecture, the internal structure. And researchers studying this in detail have discovered something specific, more precise than simply fewer slow waves. It's the rhythm of those waves that breaks down. In healthy young adults, slow waves occur in what researchers call oscillation trains, clusters of consecutive waves, one after another, maintaining a sustained pulsing rhythm, like a river, continuous, directional, efficient. That sustained rhythm is what drives the cerebrospinal fluid exchange that makes glymphatic clearance possible. In adults over 60, those trains grow shorter, more isolated. The brain enters slow-wave sleep, generates 2 or 3 waves, then fragments, producing individual isolated waves rather than sustained oscillatory sequences. Think of it this way: a sustained train of slow waves is a river. The pressure and direction drive efficient fluid movement. An isolated wave here and there is a dripping faucet. The water still moves, but the pressure that drives meaningful clearance through dense brain tissue is gone. A 2025 randomized crossover trial published in medRxiv with a multi-site design involving 39 adult participants measured beta-amyloid and tau directly in participants' blood plasma before and after a full night of sleep versus after sleep deprivation. They combined EEG measurement, pulse transit time tracking for cerebrovascular compliance, and transcranial impedance spectroscopy to monitor glymphatic flow in real time. The result was clear. The duration of NREM sleep, specifically stages N2 and N3 combined, was the strongest single predictor of overnight beta-amyloid and tau clearance from the brain to the plasma. Not total sleep time, not subjective sleep quality, not REM sleep, NREM sleep duration. A separate study, published in Molecular Psychiatry in 2025, analyzing 72 older adults using both MRI and full polysomnographic sleep recording, found that the DTI-ALPS index, the current best non-invasive proxy for glymphatic function in living humans, was negatively correlated with sleep quality scores and apnea-hypopnea index. The worse the sleep quality in older adults, the lower the measurable efficiency of the brain's cleaning system, consistently, across individuals, across imaging modalities. Now, before we go to the solutions, I want to give you something that most health content leaves out, the honest version of where the science stands. There's an active debate in neuroscience right now about whether glymphatic clearance happens exclusively during sleep or whether some of it also occurs during wakefulness through partially different mechanisms. At the 2025 Sleep Annual Meeting, researchers from Mount Sinai's Icahn School of Medicine presented data suggesting that clearance may also occur efficiently in certain waking states. This is real science. I'm not going to pretend the picture is simpler than it is. What the current weight of evidence strongly supports is this: deep NREM sleep remains the primary, best documented, and most controllable window for glymphatic activity.
It is the window that declines most sharply after 60, and it is the window where targeted interventions show the clearest measurable effects. That's where we go next. 3 biological interventions, each with clinical evidence, each targeting a different part of the problem. I want to start by telling you what these are not. They are not sleep hygiene tips. They are not avoid your phone before bed. That advice exists for a reason, but it operates at the surface. What I'm going to describe operates at the level of physiology, at the level of what your hypothalamus, your glial cells, and your synaptic receptors actually need to generate sustained, slow-wave architecture. The first and most immediately actionable is body temperature. Here is the mechanism. Your hypothalamus monitors your core body temperature as one of the primary signals for sleep stage progression. To initiate and sustain N3, deep slow-wave sleep, your core temperature must drop by approximately 1 to 1.5 degrees Celsius from its baseline. This is not optional. It is a physiological requirement embedded in your circadian biology. When core temperature remains elevated from a warm bedroom, from eating a large meal close to sleep, from certain medications with thermogenic effects, the hypothalamic signal that allows full entry into N3 is weakened. You get lighter sleep, more fragmentation, fewer oscillation trains. The clinical target for bedroom temperature is 65 to 68 degrees Fahrenheit. 18 to 20 degrees Celsius. This is not a preference. This is the range consistently associated with optimal slow-wave architecture across adult populations. Here's the counterintuitive intervention. One of the most effective ways to lower your core temperature before bed is to raise your skin temperature first. A warm bath or shower taken 60 to 90 minutes before sleep triggers a process called vasodilation. Your blood vessels near the skin surface dilate, drawing heat from the body's core outward. You step out of the bath. The skin surface cools rapidly. The core follows, dropping faster than it would have otherwise. Your hypothalamus registers the drop. The signal for slow-wave sleep fires earlier and more strongly. A study monitoring 50 community-dwelling older adults over 18 months, more than 11,000 person nights of tracking, found small deviations from the optimal bedroom temperature range disrupted sleep quality to a degree comparable to the effects of chronic pain or late-night alcohol consumption. For older adults who are already generating fewer sustained oscillation trains, that additional disruption matters disproportionately. The second intervention is magnesium, and the specific form of magnesium matters enormously here because most forms you find in pharmacies do not cross the blood-brain barrier in concentrations sufficient to affect sleep architecture. Magnesium threonate, also known commercially as Magtine, is the exception. It was specifically engineered to penetrate the central nervous system, and its mechanism for supporting deep sleep is distinct from simple relaxation. Magnesium modulates two critical receptor systems in the brain: GABA receptors, which promote inhibitory signaling and reduce neural arousal, and NMDA receptors, which, when chronically overactivated, contribute to the hyperexcitability that fragments sleep in older adults. In simpler terms, magnesium doesn't sedate you. It removes the neurological resistance that keeps your brain from descending into deep sleep when it should. There's also a population-level issue worth naming directly. Research suggests that a significant percentage of adults over 60 have insufficient magnesium intake, not because of a dramatic deficiency, but because absorption declines with age, and dietary sources alone often fall short. This matters for sleep because even a modest functional deficit in magnesium can chronically upregulate NMDA receptor activity, keeping the brain in a low-grade state of arousal that prevents the full expression of slow-wave oscillation trains. It is not a dramatic problem, it's a quiet one, and quiet problems in sleep architecture accumulate over years, not overnight. A study published in Sleep Medicine X in August 2024, 76 adults with self-reported sleep problems, tracked over 21 days using Oura Ring wearable data, found statistically significant improvements in objective deep sleep duration and REM sleep duration in the magnesium threonate group compared to placebo.
Daytime alertness and mood scores also improved significantly. A second trial, published in Frontiers in Nutrition in January 2026, 100 adults, 6 weeks, randomized, double-blind, placebo-controlled, found that magnesium L-threonate was associated with a statistically significant improvement in overall cognitive performance on the NIH Total Cognition Composite, with the largest treatment effects on working and episodic memory. Researchers also calculated a 7.5-year reduction in estimated brain cognitive age compared to placebo, measured using standard neuropsychological benchmarks. Now, a necessary note before you act on this. Magnesium is cleared by the kidneys. If you have any history of kidney disease, reduced kidney function, or are on medications that affect potassium or magnesium balance, speak with your doctor before starting. This is not a disclaimer I'm adding to protect myself. It's accurate biology that matters for a meaningful percentage of adults over 60. The third intervention requires no purchase and no prescription: sleep position. Research, primarily in animal models with consistent supporting signals in human imaging studies, suggests that lateral sleeping position on your side allows for more efficient glymphatic flow. The geometry of the paravascular spaces in the lateral position appears to support more effective cerebrospinal fluid exchange than supine or prone positioning. This is not a reason to force yourself into an uncomfortable position and stay there rigidly all night. Natural position changes during sleep are normal and healthy. But if you're choosing between positions as you fall asleep, the evidence modestly favors lateral. And for adults who use only one pillow and sleep on their back by habit, simply adding a second pillow between the knees, which naturally encourages a lateral orientation of the hips and spine, can make the transition more comfortable and sustainable over time. Let me bring everything together. Your brain has a biological system that runs every night to remove the proteins most strongly linked to Alzheimer's disease. That system is most active during deep NREM sleep, a stage that declines by 50 to 80% between your 20s and your 60s. That decline is not uniform, it is not inevitable in its rate, and there are biological levers with clinical evidence behind them that directly influence how much slow-wave architecture your brain generates each night. Bedroom temperature, 65 to 68 degrees Fahrenheit. A warm bath or shower 60 to 90 minutes before sleep.
Magnesium L-threonate, the form that reaches the brain, with 2 randomized controlled trials supporting its impact on sleep architecture and cognitive function, and a conversation with your doctor if kidney function is a consideration. Lateral sleep position, low cost, 0 risk, and physiologically supported. None of this replaces medical care. None of this guarantees an outcome. What it does is give you, probably for the first time, a mechanistic understanding of what your brain requires from sleep to protect itself. And it gives you one specific, intelligent question to bring to your next appointment. Can you check whether my sleep architecture, my N3 slow wave time specifically, might be contributing to amyloid accumulation risk, and is there a reason I shouldn't try magnesium L-threonate? That question, informed by actual clinical research, is worth more than any sleep tip list you've ever read. I want to know, has your doctor ever mentioned sleep architecture, slow wave sleep, or the glymphatic system? Drop your answer in the comments below. I read every single one. In the next video in this series, we go deeper into what happens when the glymphatic system isn't just running slowly, but structurally impaired, what the latest imaging research shows about who's most at risk before a single symptom appears, and what that means for how you think about brain protection in your 60s and beyond. Don't miss it.